The process of eukaryotic transcription has similarities to bacterial transcription, but also it shows differences. The similarities includes, for example, stages such as initiation, elongation, and termination, which are similar to bacterial transcription. And there are many differences. Three of them are listed right here. First of all, as you recall, during bacterial transcription, one RNA polymerase recognizes promoter and initiate and transcription as well as play a role in elongation. Eukaryotic cell possess three different RNA polymerases. Each of them transcribes a different classes of RNA and recognizes a different type of promoter. Secondly, in eukaryotes, it is not RNA polymerase that recognizes promoter. It is a complex of protein, which we call accessory proteins, which binds to promoter, to consensus sequences of promoter. And upon assembly of this protein, accessory protein complex, RNA polymerase is called in. Thirdly, as you recall, in eukaryote, DNA is wrapped around the histones. So chromatin structure has to be modified in order for polymerases to gain access to DNA. The most common modification, or the best studied modification, is acetylation of histones. Acetyltransferases add acetyl group to histone tails and destabilize nucleosome structure. How and why? Do you remember? Histone tails have positive charge. DNA, through the phosphate group, exerts negative charge. So this positive and negative charge keeps histone and DNA in close interactions. Once you decrease positive charge on histone tails, the interaction between DNA and histone loosens. Another way how chromatin can be modified is through chromatin remodeling proteins, which can actually bind to chromatin and displace nucleosome from promoters or other important regions. As you recall, eukaryotic transcription system contains three different RNA polymerases which transcribe different classes of RNA and recognizes a different type of promoter. Here we will focus predominantly on RNA polymerase II, which transcribes genes that encode proteins, and promoter for a gene transcribed by this polymerase consists primarily of two parts: the core promoter and regulatory promoter. The core promoter is the place where basal transcription apparatus and RNA polymerase assembles. It has two consensus sequences. First one at position -25, it can go from -20 to -30, is so-called Tata box. Tata box you find in every eukaryotic gene core promoter. Tata box is place where TBP binding proteins binds and start to unwind DNA. In addition, in position of -35, there is error, it's at -35 base pair, there is TF2B recognition element or consensus sequence where general transcription factor TF2B binds. So, to summarize, core promoter is upstream of initiation of transcription, which initiates at the so-called initiator elements, and it has kata box at -25 and TF2B recognition element at -35 position. In addition to core promoters, genes which are transcribed by RNA polymerase II usually have regulatory promoter. Regulatory promoter is located upstream of core promoter and contains different consensus sequences. Usually it contains mixes and matches of different combination of consensus sequences to which different transcription factor binds. So regulatory promoter is very specific for each particular gene, and therefore there is not common structure or common consensus sequences. It's more or like the different gene combine different consensus sequences and use different transcription factor to enhance its transcription. In some genes you can also find enhancers. Enhancers can be upstream located or downstream located from core or regulatory promoter. An enhancer can be close to core promoter, but they can also be far apart, sometimes even 1,000 base pair apart. Now, RNA polymerases 1 and 3, each one recognizes different promoters and one distinct from that one recognized by RNA polymerase 2. For example, they recognize promoter for small ribosomal RNA, tRNA genes, and so on. Transcription in eukaryotic cells is initiated when regulatory proteins assemble and bind to DNA near the promoter and start to modify chromatin structure and the transcription site. So what happened first? We will focus on the general transcription factor TF2D, which associates with several polyproteins, and one of them is so-called Tata binding protein or TBP. So when TBP binds to the DNA, it actually bends the DNA and trigger partial unwinding. This partial unwinding actually allows additional transcription factor, and RNA polymerase binds to the core promoter right here. The additional transcription factor, you remember TF2B binding site on the promoter. So there are additional transcription factor, basal transcription factor, TF2 beside TF2D, TF2B, TF2A, TF2H, F, and E. So this general transcription factors assembles and attracts RNA polymerase right here. And basically once this complex of transcription factors and RNA polymerase assembles, it actually triggers further conformational change, which trigger unwinding of the 11 to 15 base pair of DNA which surround transcription start site, and we call this so-called open complex. Once the open complex is present, RNA polymerase start to transcribe. Now, as we mentioned, this basal transcription apparatus and RNA polymerase are necessary to initiate transcription. However, they are not sufficient to accomplish successful fast transcription of the genes. We do need transcription factors which bind to regulatory promoters. These transcription factors which binds to regulatory promoter through the mediator and co-activators interact with basal transcription apparatus and speed up and enhance transcription. Similar applies to enhancers. Enhancers can actually attract the protein, enhancer proteins, which actually would trigger bending of DNA right here. DNA creates a loop, and through the coactivator, through the mediators or coactivators, it can actually interact with basal transcription apparatus and initiate transcription. Once RNA polymerase synthesizes approximately 30 base pair of RNA, the RNA polymerase leaves the promoter and enters elongation stage of transcription. In this picture, you can see the structure of the RNA polymerase II. Sometimes we call it also Kornberg enzyme, and it's because its structure and function has been revealed by Roger Kornberg, who was awarded Nobel Prize for chemistry for this discovery. Now, the DNA double helix enters through the groove here into the enzyme core. It's unwound, and the nucleotides are added according to the template sequence, and this nucleotide enters through this pore, are funneled in through the pore. Once the RNA is synthesized on the DNA template, it's separated from DNA and exits through another groove, and DNA is rewound together. The three RNA polymerases use different mechanisms to terminate transcription. RNA polymerase 1 requires a termination factor which is similar to raw protein in bacteria. However, it does not bind to RNA as in bacteria, it does bind to DNA downstream of the termination site. RNA polymerase 3 ends transcription after transcribing a terminator sequence that produces a string of uracil. It does not require hairpin structure. That string of uracil destabilizes DNA-RNA hybrids and releases RNA. RNA polymerase 2, which we focus predominantly at, requires a termination enzyme RAT1, and we will talk about it in next slide. Let's now talk about transcription termination during RNA polymerase II transcription. The RNA polymerase II continues to synthesize RNA hundreds, even sometimes thousands of nucleotides beyond the coding sequence, which is necessary to produce the mRNA. Now, at the 3' of this transcribed sequence, there is consensus sequence which defines cleavage site. At this cleavage site, RNA is cleaved and yields 5 to 3' mRNA and 5' end short RNA which has 5' trailing end. This 5' trailing end of RNA is recognized by RAD1 exonuclease, and as it binds to this trailing 5' end, it starts to digest RNA. It basically chews it up until it reaches transcription apparatus. Once it reaches transcription apparatus, the transcription is terminated and transcription apparatus is disassembled, and you will have pre-mRNA transcribed right here.